We've seen how we can use a t-test to compare two means, such as with Alice's comparison of a control and a treatment group. But what if you have more than two groups to compare? As an example, suppose we are looking at factors affecting the growth of stem cells. Stem cells are important for a range of applications, but cell survival is often low. Aiming to improve this, researchers investigated growing cells inside fibrin scaffolds. Mouse embryonic stem cells were cultured in 16 wells seeded with fibrin scaffolds. With at least 5,000 cells in each well, fluorescence-activated cell sorting was used after 3 days to count the number of living and dead cells in each well. This plot shows the survival data from the 16 wells. There is quite a lot of variability, with cell survival less than 40% in some wells and more than 80% in others. If nothing else is known about the experiment, then the best we can do with these data is to report the sample mean, shown in red, and the standard deviation around the sample mean. Again, there is quite a lot of variability about that mean. The sample standard deviation of these 16 observations is 16.81%. Recall that this value comes from the sample variance, the sum of the squared deviations from the mean divided by the degrees of freedom, n-1. Here that sample variance is 282.52. The units are percentage points squared. Knowing this, we can rearrange the formula to figure out that the sum of the squared deviations is 4,237.8. We will refer to the overall variability in the response as the total variability. Our measure of this total variability is built from these two parts, the total degrees of freedom, DFT, and the total sum of squares, SST. Note that when we talk about a sum of squares in statistics, it is usually around some estimate. The ratio of a sum of squares to its degrees of freedom is called the mean sum of squares, written as MS. Note that this is really just another name for a variance. Of course, there was more to this experiment than simply studying the variability in cell survival rates. The experiment was actually conducted at 4 different concentrations of neurotrophin-3, a protein growth factor, with 4 replicate wells for each concentration. This figure shows the results, now split by NT3 concentration. The cell survival values are the same as before, but now there seems to be less variability within each NT3 group, since it appears there are differences between the NT3 groups. We can summarize the differences by working out the mean cell survival in each group. If we wanted to compare just 2 of these groups, we could use a t-test. But how can we give an overall assessment of an NT3 effect? After calculating a separate mean for each treatment group, we can also calculate the standard deviation of the variability within each group. As with the total variability, each of these standard deviations came from a sum of squared deviations divided by degrees of freedom. Here are the corresponding values for this data. To proceed with this analysis, we're now going to make the assumption that the underlying population standard deviation for cell survival is actually the same for each concentration group. This is similar to the assumption we made for a pooled 2-sample t-test. Of course, we'll need to check that this assumption is plausible, given the observed sample standard deviations. However, if it is plausible, it means that a squared deviation from the mean in the 0 concentration group is estimating the same variability as a squared deviation in the 10 concentration group. This allows us to pool together all our estimates across the 4 groups. This gives us a pooled sum of squared deviations of 1458.6. We call this the residual sum of squares, or SSR, since it is the variability left over after adding our different means for the NT3 groups. We similarly pool the degrees of freedom, so DFR is 12. In general, we lose 1 degree of freedom for each sample mean we have to estimate, so if we had K groups, then we'd have n minus k residual degrees of freedom. The overall sum of squares was 4238, but after fitting the 4 levels of NT3 concentration, the residual sum of squares is only 1459. This suggests that knowing the NT3 concentration has given us some information about cell survival, as we already saw graphically. We calculate that the sum of squares explained by the 4 groups is 2779. We can attribute this measure of variability to a possible relationship between cell survival and NT3 concentration. This estimate has 3 degrees of freedom. A useful statistic for our model of cell survival is the R-squared value. This gives the proportion of the total sum of squares that is explained by the factor of interest. Here we see that knowing the level of NT3 concentration explains about 66% of the overall variability in observed cell survival. R-squared is particularly useful for comparing the quality of models, but to test for a significant effect of NT3 concentration, we use a different statistic. To see how this works, here is another example where the null hypothesis is actually true, and there is no difference between the 4 groups. This time, there doesn't seem to be much change in the variability on the right compared to the overall variability on the left. Indeed, we find that the residual sum of squares is much the same as the total sum of squares. If there really was no association, then we would expect these sums of squares to be similar, giving roughly 0 for the group sum of squares. However, even if there is no association, we would never see exactly 0 because of the natural variability in cell survival. That is, the sum of squares has a sampling distribution, as it will change from sample to sample. So let mu0, mu10, mu20 and mu30 be the underlying mean cell survival proportions for the 4 NT3 concentrations. Our null hypothesis is that all 4 of these means are equal, that there is no effect of NT3 concentration on cell survival. The alternative is just that the means are not all equal. This is a very general alternative. For example, it could be that all 4 means are different from each other, but it could be that 10, 20 and 30 are all the same, but are together different to 0. If there was no association, we would expect the variance between the 4 groups to be similar in magnitude to the variance within the 4 groups, the residual variance. This gives us the F statistic, which is a ratio of the mean sum of squares for the groups to the mean sum of squares for the residuals. Recall that a mean sum of squares is just a variance, so the F statistic is a ratio of variances. That is why this approach to comparing groups is known as the analysis of variance. For our cell survival study, we find an F ratio of 7.62. That is, the variability between the groups is 7.6 times more than the variability within the groups. If the null hypothesis is true, then this statistic has an F distribution. Note that there are 2 degrees of freedom involved in this statistic, since there are 2 variances in the ratio. This is the probability density function for the F312 distribution. Since it is all about sums of squares, you can never get a negative value for F. But if the residual variability was close to 0, then you could have a very large value for F. As a result, the F distribution is skewed to the right, unlike the normal distribution and T distribution. From this figure, it seems like an F value of 7.62 is pretty unlikely. From tables or software, we find that the p-value is less than 0.01. This gives strong evidence against the null hypothesis of equal means, suggesting that NT3 concentration does have an effect on cell survival. This procedure is an example of analysis of variance, usually abbreviated as Anova. The results are traditionally summarized in an Anova table. This table shows all the steps involved to get to the p-value, but it is also a useful summary of the experimental design. For example, since the total degrees of freedom are 15, we know there were 16 experimental units. With 3 degrees of freedom for NT3, we know that there were 4 groups being compared, and so on. The advantage of analysis of variance and the F-test is that they are very general methods. Note that the Anova table doesn't explicitly mention the sample means at all. We have been able to infer that the population means are different by reasoning about variance. This is very cool, but why couldn't we just do lots of 2-sample t-tests to compare the different groups? Well, here we'd have to compare 0 NT3 against 10 NT3, 0 NT3 against 20 NT3, and so on, a total of 6 comparisons. That is a lot of work to do by hand, but R wouldn't mind it. The main reason is actually that every time we do one of these tests, we expose ourselves to making a type 1 error. If we do lots of tests, then our chance of rejecting a null hypothesis that is actually true can become very high. The Anova F-test protects us by doing a single test to see if there are any differences. If the answer is yes, then we can look at multiple post hoc comparisons to see where those differences might lie.